Okay, so in the previous lesson, we talked about cholera and its prevention and treatment. So in this class, we'll talk about malaria and then we'll also talk about AIDS. So to start off malaria, we'll talk about the pathogen. So the pathogen which spreads malaria is Plasmodium, and it has four variants, which is Falciparum, Vivax, Ovale, and Malariae. The type of organism, it is a protozoa, which is a eukaryote, and that is important when we talk about why it's difficult to make vaccines for malaria. But for now, we'll just continue. So transmission is by female mosquitoes, as we know. So the insect vector, so the transmission is by insect vector, and the specific mosquito is the Anopheles mosquito. You can also obtain plasmodium through blood transfusion using unsterile needles, also through mother to child due to blood across placenta. So it's supposed to be child here, there's just a missing word. Also, when we talk about inside the vector itself, we know that the female Anopheles mosquito needs the human blood to obtain proteins which are required to make eggs. And when the person is infected, the plasmodium parasite can enter from the human into the mosquito during the blood meal. And what happens is that the gametes of the parasite, it will fuse in the gut, and the infective stage will basically move to the salivary glands. So it needs to move to the salivary glands because near the salivary glands, it's where the blood meal happens, right? So that's where they take the blood in. So that's where the exchange will occur. Then what will happen is that the mosquito will take its next blood meal, and then the infective stage will obviously leave the salivary glands, and then they'll enter the human blood. So once it leaves the mosquito and enters the human, they can either enter the red blood cell or liver cell, and within the cell, they will multiply. And we call this antigen concealment because the antigens in on the plasmodium itself are not exposed to white blood cells. So starting an immune response will be difficult. And also, what happens is that once they multiply, the the cells get damaged and then it will burst, causing the plasmodium to enter the blood and spread more. So, as we talked about the site of action, which is red blood cells, liver cells, and brain. So, the symptoms of malaria is spleen enlargement and fever. So, spleen enlargement is due to a lot of red blood cells being damaged during um, the plasmodium multiplying. So, what happens is that the damaged red blood cell needs to be destroyed. The treatment of malaria usually involves using of anti-malarial drugs, which are example chloroquine or hydroxychloroquine. For example, mefloquine as well. But the issue with mefloquine is that it's expensive and unpleasant side effects. And these anti-malarial drugs, they can act on the parasite by inhibiting sexual reproduction or inhibiting protein synthesis. You can also use some antibiotics like doxycycline. So I'll recommend you remember one or two of each example so you could quote them during the exam. And also the dipstick test is used for fast diagnosis. Um, to prevent malaria, so what you need to do is get rid of stagnant water. So stagnant water allows the larvae to um, grow up and also allows them to give birth in the stagnant water. So you can treat marshes or clear vegetation. You can also cover the surface of water with oil, so which will cause the larvae to die. Introducing gambusia fish and a bacterium which is known as Bacillus thuringiensis to the water, it'll cause they feed on the larvae. So what happens is in point 2 and 3, you're reducing the potential insect vector. So you're reducing the number of potential mosquitoes which can act as carriers for plasmodium. And when you use the bacterium Bacillus thuringiensis, it's not toxic to other life forms. It will only attack the larvae. Also, you can talk about individual responsibilities such as reducing exposure of skin at dusk. Also, you can use mosquito nets which are soaked in insect repellent. You can also use prophylactic drugs. Prophylactic drugs are drugs that must be administered before you enter a country where the disease is present or endemic. Furthermore, the genome of the plasmodium is fully sequenced, so this allows multiple vaccine, or sorry, not multiple research facilities to lead the development of vaccinations and stuff. So, so let's talk about the difficulty in developing vaccines. So we know that the entire genome is sequenced. So what's the problem with developing a vaccine for malaria? So the first issue is that there's multiple stages of development of the parasite. So the antigens keep changing in each parasite in each life cycle. So there's one in the mosquito, there's one in the human. The antigens on the parasite are concealed. So what happens is that the parasite will enter liver and red blood cells to multiply. So obviously white blood cells can't come and bind to these protein receptors to get activated and stimulate an immune response. There's multiple strains of the Plasmodium. They have also drug resistance, resistant to DDT and other insecticides. Furthermore, there's also migration, which is increasing nowadays because of economic and political reasons, and climate change and environmental change. So this causes more rain, increase in temperature. These are all favorable factors for spread of mosquito. Global distribution is basically that they are mostly found in tropical and subtropical regions, just because it provides the critical factors, which is warm hot temperatures and also water or rain, which is required for the larvae. So AIDS is basically acquired immune deficiency syndrome, and it's caused by the human immunodeficiency virus. The pathogen is the HIV, and it's a retrovirus, which means that it contains RNA as a genetic material, which which is uncommon. Like as we have only been talking, the DNA is the genetic material. So a retrovirus contains RNA, and it has enzymes which are reverse transcriptase and integrase. So we'll talk about that. Those are important enzymes. So what happens is during transmission, so we know that this is due to intimate human contact, where there's exchange of bodily fluids during sexual intercourse, sharing of intravenous needles, blood transfusions, placenta and breast milk, mother to child, also mixture of blood during birth. Furthermore, you can also increase your risk with multiple partners. This is in relation with point one. Okay, so within the body, what will happen is that the membrane of the virus will, and the host will fuse, which allows the enzyme reverse transcriptase and enzyme integrase and viral RNA to enter the cell. So viruses are parasites which depend on the host cell for multiplying. So what happens is that the reverse transcriptase will convert the RNA to DNA, and integrase will incorporate the viral DNA into the host cell. So what happens is that this transcription and translation of the viral RNA or viral DNA being taken place. So the viral RNA and proteins will basically spread from cell to cell. Furthermore, so you know that the site of action is T helper lymphocytes, macrophages, or brain cells. Symptoms of HIV there isn't much, but AIDS they are associated with opportunistic diseases, because eventually AIDS is just a lack of immune related cells. So this the immune system is weakened. So we know that there's no treatment for AIDS, but there's a drug therapy which can slow the onset of AIDS. There's a specific drug which is known as xenobutine, which has a similar structure to the nucleotide thymine. So what happens is that they can bind to this enzyme known as reverse transcriptase. We were talking about it, and blocks its action. So it prevents replication of the genetic material from taking place. So obviously slowing down the onset of AIDS. These are expensive drugs. It's a long-term treatment. So these are all factors which make it unaffordable in third world countries. The drugs they have unpleasant side effects which can be permanent, and also severe ones like nerve damage, and also temporary mild ones like headaches. So the prevention, it's difficult because it has antigenic shift. So it's difficult to recognize and control. Antigenic shift is when the proteins um, on the virus's cell surface membrane they change. So it's difficult for the immune system to bind to them and recognize them. You can educate the public or young children with about the spread of infection. You could also use condoms, dental dams, which prevent exact exchange of bodily fluids. Contact tracing is important, which is what happens is contact tracing is that if one person is infected, you ask them to list their sexual partners, and then you go and check with all of them, and then you check their sexual partners. So contact tracing. You can also discourage drug use, or you can distribute the needles itself. So you exchange use one for sterile one. So what happens is that there's no sharing being taken place. You could also screen. Obviously, you have to screen the blood. This happens nowadays that you screen the blood before you um, use it for blood donation. You could also, importantly, you have to check for sex workers as well, because they're obviously the most affected by such diseases. Also, in high income countries, it's more encouraged to discourage. Sorry, it's better to discourage breastfeeding, but in low income or middle income countries, basically what will happen is that they um, advise breastfeeding, because this gives immunity to certain diseases, because there's also a lack of like clean water to make formula milk. So it outweighs the risk of getting HIV. So we'll talk about TB and other related stuff in this chapter in the next few videos. So I hope you found um, these shorter videos more helpful in understanding more about malaria and AIDS and their prevention and treatment and such. So thank you for attending the lesson and hope you understood everything. If you have any questions, leave them in the comment section. I'll see you in the next lesson. Thank you.